So today I'm going to try to make the case that not only did you do research this summer, but you learned lessons that maybe you are not aware of that are going to make you better doctors. Throughout my life, in rounding with people, I've always noticed that physicians who've had any research experience end up having a different approach to medical care, and I'm going to try to make that point today. So my main goal today is to focus on attitudes. We're not talking about knowledge, we're not talking about skills. It's really about your attitude towards clinical medicine and science. What do scientists have to offer? You've been surrounded by this summer, you already have been indoctrinated into the scientific culture, and I'm going to drill down and give you some very specific examples. First, I want to pose the problems that we have in medicine, and God willing, your generation will fix these. We have medical error. You've probably heard this from other faculty, is the third leading cause of death in the U.S. It's cancer, heart disease, medical error. It surpasses all those other diseases that are important to us, Alzheimer's disease, neurological diseases, GI diseases. Third leading cause of death, which also as an aside, if any of your family ever have to be in the hospital, I recommend that you're there by their bedside 24/7. The next problem is unnecessary medical costs. The Institute of Medicine has guesstimated that $200 billion of healthcare spending is for unnecessary treatment. That's a staggering figure, and largely the problem is due to the way we approach interventions. So we're going to talk about that today, and then I'm going to finish up with adverse drug events. Adverse drug events cause thousands of deaths per year. You've seen every TV show has some pharma ad on some drug. They mumble the side effects that you can't even hear what they are, and then you end up, asking your doctor for the drug because the advertisement has worked. So we're going to work on those three topics. So the causes of medical error are many. There's medication related to anesthesia, infections, missed or delayed diagnosis, avoidable delay in treatment, inadequate follow-up after treatment, inadequate monitoring after a procedure. But today we're going to focus on missed or delayed diagnosis. 30% of medical error is missed or delayed diagnosis. It's either missed, it's wrong, or it's delayed. And the question is, does thinking scientifically improve diagnosis? So let's talk about reducing diagnostic error. Scientists and yourselves engage in what we call hypothetico-deductive thinking. You have a hypothesis based on all your readings. You have an idea, hypothesis. And then you make some deductions about that. Well, if that's true, I'm going to do that experiment because it's going to show X, Y, and Z. That's really the basis of great diagnostic reasoning. Scientists also seek direct evidence. I'm sure you heard that term in your labs. What's indirect evidence is useless to a scientist because it really ends up not proving what they set out to prove. They look for direct evidence. They also consider all possibilities. They don't just say, well, I'm going to focus on these things, even though the literature says all this stuff. They focus on all possibilities, and they set out specifically, I'm sure you had these discussions, to prove the other hypotheses wrong, because that strengthens your own hypothesis. You say, well, it could be this finding, but I'm going to show that that's wrong by doing this experiment. All four of these will make you a better diagnostician. So let's start. You had cardiology, right? Good. So you're whizzes at this, and you had kidney. Patient presents with edema, and through induction, you come up with three diagnoses: heart failure, cirrhosis, and nephrotic syndrome. And now you make a deduction. If you do a measure ejection fraction, that will help you decide if it's heart failure. If you measure albumin in the plasma, that will help you decide if it's cirrhosis or nephrotic syndrome. And you make the measurement, and the albumin is low, but the ejection fraction is normal. So that limits your possibilities, and now you focus on cirrhosis and nephrotic syndrome, and now through deduction, if it's nephrotic syndrome, there should be protein in the urine. And if there isn't, then it's not likely to be nephrotic syndrome. This is classic hypothetico-deductive reasoning. So why is this important in diagnosis? And during your M&P sessions, you're going to have sessions with Dr. Nickerson, who's a clerkship director, and Dr. Cheng, who's the clerkship director of primary care. They're going to sit with you during your third year M&P sessions, And you're going to talk about diagnostic reasoning, and there are really two kinds. There's hypothetical-deductive, which we just went through. You thought through it systematically. And then there's pattern recognition. That is classically what more senior physicians engage in. Oh, I see those 3 signs and symptoms. Bingo, it's nephrotic syndrome. The problem with that is it's unsystematic and it's far less accurate. You'll get a kick out of this, but medical students engage in hypothetical-deductive reasoning, in part because they don't know what the patterns are. But then as you go through your residency training, you end up learning what the patterns are, so you think. And then you end up using pattern recognition, which is sort of reflexive. It doesn't take any work. And the problem is that it's notoriously inaccurate. So your scientific experience this summer, where you engage in hypothetical-deductive reasoning, will make you a better diagnostician. The second point is that in all your experiments, you sought direct evidence, because indirect evidence can't rule things out, because it could be explained by this or that. So just taking this little framework here, we see that there are 2 measurements here that are indirect. The albumin is low. Well, it could be cirrhosis and nephrotic syndrome, but what about kwashiorkor or starvation? Albumin is going to be low. Or maybe they've got, some immunological disease where the albumin slips into the tissues. And the same thing with albuminuria. There are many causes of albuminuria which are not related to the kidney. But you've also got direct evidence. You've got an ejection fraction which is low. That is more or less direct evidence, although in researching this I see that that may not always be the case. And you can do a kidney biopsy. So this is where clinical judgment comes to play. You could look for direct evidence by doing a biopsy of something like a brain biopsy, but you might decide not to because that has some risk. So again, scientific thinking focuses on direct evidence. The third point is that scientists consider all probabilities. They compulsively explore any work that you do. Think of the application you filled out with your mentor, the 4-pager that you handed into Dr. Fisher. Think of all the possibilities that you considered when you looked over the literature, and that sort of led you to this one idea, but you were well aware of all those other ideas. That is not always true of diagnosis. That you're aware of some diagnoses, but then you stop thinking about it. It's a classic error in diagnosis. I'll give you a brief example. These are 4 cardinal. Anybody do a neurology project this summer? These are the 4 cardinal symptoms of myasthenia gravis. Have you had neurology? Yeah, so you've heard the disease, myasthenia gravis, or maybe not. These are the classic symptoms. This is when I was training Watson how to be a good doctor, I used to test them. I used to test the machine by giving sort of classic diagnoses to see if Watson came up with the right answer. These are the classic symptoms, and pattern recognition. If you go to a neurologist and you say those 4 things, the neurologist, the first words out of his or her mouth is going to be myasthenia gravis. So I looked at the diagnostic engines, and I'm going to say the word Isabel today, and through your course, for your experience with me over the next 4 years, every time I see you, I'm going to say the word Isabel. It's a commercially available diagnostic product that has been shown unambiguously to improve diagnosis, and nobody here uses it. More about that later when I talk about resources. So Isabel had a great differential. Myasthenia gravis is there, but so is botulism. So if you were feeling pretty cool and making a snap diagnosis using pattern recognition, you saw those absolutely classic myasthenia gravis symptoms, you might not have thought of botulism. So did Watson. So Watson and Isabel got the main diagnoses, but Isabel had others that are important. Paraneoplastic syndrome, when you have cancer, you have a neuropathy that goes along with it. And Watson had diseases that I never heard of. Fonzio-Lon disease, absolutely had no idea of what that was. I mean, it's so rare. It happens that those 4 symptoms are characteristic of that disease. It ends up being a pediatric disease, which is my excuse for why I didn't know about them. I'm not a pediatrician. But my point here is that differential is really much longer than you would think, that differential diagnosis. And once again, a scientist, you don't embark on spending money on your grant unless you know everything that's out there and you consider all the possibilities. The next point is what we call avoiding confirmation bias. Confirmation bias, sort of in a lay example, is those of you who are red thinkers, conservatives will watch Fox News. People who are blue thinkers will watch, MSNBC. You kind of watch and read the sources that feed into your picture of the world, and you don't even think about the other sources, whether or not there's any validity. We call that confirmation bias. And in medicine, it's rampant at times, and it results in a lot of extra testing. So imagine you have your patient with edema and congestive heart failure, and you measure the ejection fraction. Now, if it's normal, most people would say, this is not low cardiac output, but there are times when the caretaker will say, well, hold on a second, I've seen congestive heart failure with a normal ejection fraction. So I'm going to order some more tests that are expensive, brain natriuretic peptide and troponins. But a scientist realizes the limitations on proving the hypothesis. More important, perhaps, is to eliminate the other explanations. So this is where you say, well, hold on a second. It could be venous stasis. I'm going to thrombosis. I'm going to do an ultrasound. Oh, wait, it could be cirrhosis. I'm going to measure the albumin. It could be nephrotic syndrome. I'm going to measure the urine protein. The point is that in medicine, we engage often in confirmation bias, where we keep testing the same idea when we really should be testing other ideas to rule them out. And maybe at the end of the day, it will be CHF with a normal ejection fraction. So thinking scientifically reduces diagnostic error. You employ the hypothetico-deductive process, you seek direct evidence, you consider all the probabilities, and you rule out or falsify alternative hypotheses. This will stand you in good stead if you practice this when you're on the wards. Now let's go to the second problem. Unnecessary medical care costs $200 billion a year. It's staggering. When regular people out of medicine hear that, they absolutely can't believe it. It's almost as if they made up the figure. And I'm going to focus on two problems. The first is we actually think the intervention is effective, but it really isn't. And then, worse, is we already know the intervention is ineffective, but we do it anyway. So I'm going to drill down a little bit on this. I mean, I hope that I'm not, making you more cynical as you're sitting here going, what's he talking about? I came to medicine to help people, and he's telling us all these terrible, terrible tales. So we propose that thinking scientifically is based on seeking the highest quality evidence. And you base a new hypothesis on the highest quality evidence. No scientist is going to spend NIH money if this is the state-of-the-art hypothesis. You don't confuse association with cause and effect. I'm going to give you an example of that. And most importantly, one thing I've noticed about scientists, I had a lab for a decade or so, is that if an idea looks like it's not correct, we jettison the idea immediately. We don't spend any time on the idea. I'm sure that happened in your lab meetings. You thought of something and then you realized it was wrong and you stopped thinking about it. We don't do that in medicine. So a study that I decided to show you was written by John Ioannidis, who's now at Stanford. He's a brilliant guy. He spent his whole life proving that high-quality clinical research studies are false. So here's the example. He wrote a paper where he found studies that are highly cited, like the New England Journal of Medicine, and it's a cardiology article, and then people all over the world go, wow, so-and-so is an expert. Let's do what he's or she's doing because it's, and I'll cite it, it's very highly cited. We found that 1/6 of those studies ended up being contradicted by subsequent studies. 1 out of 6 studies that is highly cited, being broadcast all over the year, is wrong. It's the opposite to what the truth is. So if you're one of those people, I'm going to try to talk you out of being one of those people, who immediately reads a paper in the New England Journal of Medicine, which some of your residents will do in a couple of years, and then goes to the bedside and does that thing. You can remind them that 1 out of 6 chance that what he's doing is the opposite to what he or she should be doing. And then others, 1/6, the effects they found were stronger than than subsequent studies. Half of them were replicated, and a quarter of them were unchallenged. Now, this is also one of my favorite articles. This is John Ioannidis. For those of you who are math geniuses, I would love any one of you to help me understand this paper. There's a lot of math, it's statistical, but basically, because clinical studies rely on statistics, and I'll tell you more about that in a second, it ends up that you can manipulate the statistics in a way to have a positive result when it really isn't positive if you knew what you were doing. And my favorite line is originally from Mark Twain. There are three kinds of lies: lies, damn lies, and statistics. I think he was referring to political issues at the time. But then I found out that Mark Twain actually got this from Benjamin Disraeli, who was a prime minister in Britain, and then they said it was from Wentworth Dilke, who I have absolutely no idea who that is. But basically, statistics can be, cherry picked in order to prove the point. And one way to sort of discuss this is to say, how do you establish a level of proof? In your experiments you've been doing over the summer, you probably did more than one experiment to go seek the truth to the hypothesis. And so a single paper can provide adequate proof. I mean, Watson and Crick, there are all sorts of single papers that revolutionized science, and it was one paper, but there were a lot of experiments. That's not true of clinical research. Clinical research is there's one experiment, and only one experiment. And then you rely on statistical analysis to see if it's valid or not, but by itself can never be adequate proof. So that's why you never act on a single article. So as I'll talk about in a couple of weeks when I do resources, we have meta-analyses now where they take many papers and then try to see what the sum result was and whether or not the idea has any validity. So this puts us behind the eight ball when it comes to clinical research, because it means that we're relying on statistics. And statistics are, I know you'll have Walter Palmer's course in the fall. He will do biostatistics. Really pay attention. He's a brilliant teacher. It's a great course because there are classic errors in statistics that will render the result meaningless. There's an idea of regression to the mean. He'll talk about that. I'm not going to talk about it today. But the classic mistake is to confuse association with causality. Even the newspapers get it wrong. The New York Times, something is associated and think it's cause and effect. So I thought I'd give you one example, my favorite example here. So Harvard School of Public Health, about 10 years ago, press release, that's always exciting. Women who eat nuts or peanut butter regularly significantly reduce their risk for type 2 diabetes. Now, if you read that title, doesn't it sound like now you're all going to rush to your refrigerators and get a jar of peanut butter, or tell your grandmother? That's not necessarily cause and effect. In fact, you have to prove cause and effect by a randomized controlled trial or something similar, data mining. And what did they actually find? They found a correlation between peanut butter consumption and a lower incidence of diabetes. There are many reasonable alternatives. That's the problem with association. It's confounding. That's one of the Achilles heels of data mining. There's confounding. And yeah, maybe it is associated and causal, but maybe not. I mean, you could reverse it and say people who eat peanut butter are different from other people. They have a gene. If you like peanut butter, it's associated with a gene that has a lower incidence of diabetes. There are all sorts of crazy explanations you could come up, but they did not show causality. What did they find? I just thought it's hard to see the results, but what they basically showed that people who never ate peanut butter and those who ate peanuts or peanut butter more than 5 times a week, they weighed more. They weighed more than 10 pounds per 6-foot person than those who didn't eat peanuts. So they weighed more. Their BMI was higher. We know the BMI is associated with diabetes. So you haven't had your endocrine section yet. So you probably already know it from reading the paper. And their total energy consumption was almost 30% higher. They ate more. Well, why would you eat more? And be thinner? Because, there are several reasons, but one is that you actually exercise more. So it's conceivable that the group they had that didn't eat peanut butter didn't exercise and they were heavier, that's why they had a lower incidence of diabetes. The only point here is that if you can't use association to prove causality. So I just sum up the quality of evidence. That's what scientists are. You are focused on the quality of the evidence. Observations and conclusions must be confirmed by additional studies. Statistical analysis of a single study is insufficient to prove. Inherent bias, we'll talk more about that later, can sway the use of specific statistics to support a potentially fallacious conclusion. And conclusions based on associations or unambiguous cause and effect. Obviously, the ones on associations cannot be trusted. So let me work on the last point of this section, which is jettisoning disproved hypotheses. As I mentioned, there are all these practices. You will go on the wards and you will see people doing things. And they will say, well, we don't think it has any effect. And then you can ask, if you want to, you have to be tactful about this. Why are we doing it? Often there are interesting reasons. We didn't know about the new evidence. It's just the way they did it. A scientist would never adopt that approach because they're wasting time. They're wasting money. Medicine adopts ineffectual, harmful medical practices, and it was summed up in this incredibly interesting book. I was jealous when I saw this book because my whole career I wanted to write this book and never got around to it. But anyway, he's a kid. He did a much better job than I ever would have done. But he's got a whole book on medical reversal where we know this intervention doesn't work, but we keep doing it, charging for it. Money is one engine. So let's just sum up. Scientists base new hypotheses on the highest quality evidence. We don't confuse association with cause and effect. And we immediately jettison disproved hypotheses. So let me go to my last point, medication errors. This is the one that you're going to have direct contact with when you go to the wards. Even when you start doing your clinical cases in the fall, you're going to see people in the hospital who are on 25 medicines or 10 medicines. Medicines often that contradict each other. So we're going to talk about medication errors. Just to give you an idea of the scope of this. This study was done 10 years ago, and it's probably out of date, meaning that it's worse than it was 10 years ago. They did a study of a single hospital, had almost 1,000 admissions. Half had clinically significant inpatient adverse drug events. Half of them. And 9% resulted in serious harm, including death. 22% had to hang around, costing both the patient and the hospital money to be monitored. And then there were, various and sundry other problems with the adverse drug event. It's a major issue, adverse drug events, and it's in large part compounded by Pharma's engine of advertising to the patient. So we're going to talk about thinking science. How does thinking scientifically help you avoid that? Well, I have a couple ideas that I'm working on. One is you need to know what can and cannot be proved. Like when you're sitting in the lab and you think of an experiment, you may think to yourself, we're not going to be able to prove that. Or that we are definitely going to be able to show that that's true. Well, you can't and cannot prove certain things with drugs. And you can also assume that the harm is greater than reported and the efficacy is less than reported. These are the three topics I'm going to focus on. So what about being proved or disproved? You cannot prove a drug is safe because there's always an exception out there. It's like for those of you who code, you can never prove that the code works all the time because there's always some example where it won't. You cannot prove a drug is safe. And no matter what the drug companies say, no matter what people say, you cannot prove that. But you can prove that it is not safe. But you need to do experiments to prove it's not safe. So where does that get us? I don't know if you're probably in high school at the time. Merck made a drug called Vioxx. It was like a new NSAID. I know you've had Pharma. Very powerful. It didn't cause GI bleeding, which the Advil and naproxen do. And it was a big winner. They made a lot of money. And then it ended up that it increased the incidence of heart attacks. And the problem was, and I'm not even blaming Merck to some extent, they didn't try to prove the drug was safe or identify all the complications. They only wanted to show that the drug was effective. I mean, maybe that's their role. This drug works. And so the philosophy or the approach is very different if you're trying to prove it's safe or try to prove it's harmful. The Merck gives the drug to patients, observational study, and it shows that there's little cardiovascular risk. It ended up there was cardiovascular risk. Some other time I'll tell you the whole story. And it was in the New England Journal of Medicine. The editors missed the cardiovascular risk, and they didn't comment on it, but it was there in the paper. But you could imagine a different group of people setting out to prove the drug causes heart disease. They do a different set of experiments. They have a bunch of mice, they give them drug, maybe do biopsies, they look at the coronary arteries. But basically nobody is going to do that. And so that makes us dependent upon, how do we approach that? Being incredibly paranoid about adverse drug events. And here's the problem with adverse drug events. There's a time course. Statins came out. It's a lifesaver. It improves, cardiovascular risk, lowers the incidence of death in coronary artery disease. Everybody I know, my age is taking some kind of statin, and there are very few side effects because it's an observational study. Often they give it to healthy people who have no problems, and there are very few side effects. And then suddenly rhabdomyolysis started being reported. That's where the muscles disintegrate and the calcium and the phosphate that are released from muscles cause acute kidney injury and sometimes death. So, in fact, some versions were so virulent in terms of causing rhabdomyolysis that it was pulled off the market. There are all sorts of statins that never made it to the drugstores because they caused a rhabdo. Then, several years later, people noticed that it caused just a simple myopathy. Like people I knew were taking it, they suddenly lost all their energy. And I was on a strong one for a while, and I found myself walking in my garage as if I had some neurological disease, and then I self-diagnosed and said it must be the statins. But that took about 5 or 10 years. And just last month, my barber, who is a brilliant quack, physician, not really, who reads the literature, he's incredibly smart, he said to me, I'm on a statin, but I'm worried about diabetes. And I went, what are you talking about? Because I didn't know that it caused diabetes in a certain number of patients. And he said, yes. So I looked it up, and just in the last couple of years, statins have been around for 25 years, I think, 90s. In the last couple of years, it's been shown to cause diabetes in some patients. And you're entering the age of precision medicine. It's probably genetically based. Some patients will be susceptible to it, other patients won't. So the problem that you face, we all face, is there's a time course of the discovery adverse events because nobody's out there looking for the adverse event because they're trying to just prove that it works. Benefits overstated, risks understated. This is a book, even though it looks like a pill box, by Ben Goldacre, who is my hero. I love preparing talks because I always discover something I didn't know. And he's written a whole bunch of stuff in the British Medical Journal, including how drug companies mislead doctors and harm patients. I mean, as soon as you set foot on the wards, you're going to be bombarded by biasing influences. At some places in this hospital, they still have pharma-sponsored lunches. I was horrified. You're going to hear a lecture or heard a lecture by David Rothman. They're buying lunches for people, which have been shown for 30 years to bias their drug prescribing. So they've worked on us, and they're really good. Pharma, they publish things, but they cherry pick the results. The Merck story is actually particularly disturbing because not only did they cherry pick the results, but they also dropped patients out of the study who had heart disease because it was going to ruin, it was going to actually show that it caused heart disease. And then late-breaking meetings. There are all these, I don't know, maybe some of you will go to a meeting based on your summer research. There are these late-breaking meetings, and people get up there, and they talk about this drug or that drug. Forget about it. It's more likely to be wrong than it is to be right. And also, you're not going to know what the side effects are. So what can you do to avoid adverse events? You know what has been proved and what hasn't been proved. Assume that harm is greater than reported. That's a given. And it'll make you a better prescriber because you're going to say, well, hold on a second. This drug is said to be harmless. That can't possibly be right. And also assume the efficacy. See, drug companies are doing all the studies. They're going to tell you the drug works better than it actually does work. And in some cases, the drugs are worthless. They have almost no efficacy, but they're advertised on TV. You can tell I'm paranoid and passionate about this. So let me end the talk and focus on attitudes. As I said in the beginning, this talk was to raise the issue of attitudes. Physicians are not trained to be skeptical. That's really the word. When you leave today, when you see me in 5 years, like at some airport, we bump into each other. We have this family joke that whenever we go on vacation, we could be anywhere, in Istanbul or Morocco, I will bump into a PNS student who was in one of my talks, and we will talk. So when I bump into you, 5 or 10 years, I want to hear the word skeptical come out of your mouth, because that's really what we're talking about. But we're not trained to be skeptical. We want to believe. We have people who are sick. They're in front of us. Of course we we want to believe that we can make them better. Not for any other reason than we wanted to make them better. I'm sure you probably put this in your essay for your medical school applications in some form. And sometimes we have to choose the best option available when there really is no good option. So we'll choose something that we think is better than nothing, but even that hasn't been tested. Maybe it's not better than nothing. Maybe it's worse than nothing. A lot of the interventions in the electronic health record ended up being worse than nothing. They actually made things worse, unintended consequences. And I think our optimism is justified. I mean, you don't want a grumpy physician to come in your room and go, I don't think you're going to get better, and just be all down. You want someone who's really enthusiastic and want to heal you, even if reliable therapy is unavailable. Scientists have the opposite philosophy, the opposite attitude, and that's what I'm hoping you gain this summer. They learn to be skeptical. They assume that there's another explanation for the thing that they've identified, and that, I'm sure, expands the differential diagnosis. You're going to go on the wards, and the patient will come in, and there'll be three diagnoses in the differential diagnosis, and you go, hold on a second, what about that Faziolan disease? Maybe it's that, and you might be ridiculed for that. Often the attendings say to the students, don't give me a list of 15 things, it's obviously 1, 2, and 3. Humor them, but you reserve the right to keep those 15 things on your list, because even if there's a 5% chance that the patient has a disease, it means 1 out of 20. It means on the 20th person who comes through the door, they may actually have that really low probability illness. You want to rule out alternative explanations. When you work up a patient, you want to not confirm over and over again your favorite diagnosis. We have anchoring bias in medicine. I'll talk to you about that in a couple of years, where we get glued to some idea we're anchored to, we just can't give it up. And that makes us fall prey to confirmation bias. We keep saying, well, it's this, but it's an unusual manifestation of this. Let's get tests to confirm it, when in fact you should be ruling out the other hypotheses just the way you did this summer. And be aware of biases out there looming that will influence the way you interpret results. There are several biases that we'll go into later on that can actually make you have your heart set on this result, or you're a scientist who just spent $500,000 on this experiment and it's really not showing what you want. So you're going to end up interpreting the data differently, and you will be aware of that. And lastly, know what can and cannot be proved. Is a drug ever safe? No. So I want to end with a couple of quotes. This is Peter Medawar, who I think you had immunology. He's the guy basically who invented transplant biology and the immunity and that. He won a Nobel Prize. And he said, the scientist is preeminently a man or woman who requires evidence before he or she delivers an opinion. And when it comes to evidence, he or she is hard to please. So you want evidence, and you're going to be really picky about what that evidence is. That's, I think, what you learned this summer. And it doesn't happen in the clinical space. Here are two more briefly. Mike Bishop is an MD, PhD. He also won a Nobel Prize for something, I can't remember what. The physician needs knowledge, critical judgment, and restraint, qualities all inherent in the ethos of science. Restraint. It's going to be very hard to act like this on the wards. It's all, people running around wanting to do the right thing, but often acting without sufficient evidence. And Frank Gannon had a good one. We are experts in our area of research, but our job demands that we constantly remain skeptical of our latest results, even when we deeply believe that we have folded new layers of truth. We behave as if the truth uncovered in an experiment might be a mirage. So when I had my lab, I'd come home, say to my wife, I can't believe the result we had today. And we'd be high-fiving each other and dancing around the table. And we'd wake up in the morning having our coffee, and she'd say, so you excited about going in, or you had that result? And I had this glum face, because somewhere in the middle of the night, I thought of 3 reasons why we saw what we saw that had nothing to do with what we were trying to prove. Alternative hypotheses. So then I had to do the experiments to show they weren't true. We don't do enough of that. So I want you to take the skeptical oath. I promise to be skeptical. I promise to assume that there are more diagnostic possibilities that I can think of. I promise to assume that clinical evidence is false rather than true. Now, I promise to assume that the benefits of a drug are less than advertised and the risks are greater. I promise that my fault is I don't believe rather than I believe. Now this will take some tact. When you're making rounds, you can't turn to attending and say, well, I don't believe that, because that's a guarantee bad recommendation or whatever it's called. So be tactful. You might, but you can think that thought. So I hope I've convinced you that your summer experience not only gave you a window into how research is performed, but at some level, hopefully, it made you more skeptical. Thank you. I'll take questions if you have any.